of the session, which it's just been a really lovely week of talks here at the Spring Symposium. But we're going to finish off with a bang. So the next speaker is Gaia Pigino from the Max Planck Institute for Molecular and Cell Biology and Genetics in Dresden. So Gaia received her PhD in evolutionary biology from the University of Siena in Italy in 2007. She moved then for her postdoctoral work at ETH Zurich and the Paul Scherrer Institute for, and she's currently at the Max Planck, but next year in 2021, she will be moving to start a new laboratory and the Human Technopole in Milano as an associate head of the Structural Biology Center that is being assembled there with some very nice equipment and other great scientists. So today she's going to talk to us about the mechanistic and towards a mechanistic understanding of ciliary assembly by correlative light electron microscopy and cryo-electron tomography. So if Gaia, if you could share your screen with us.
Yes, thank you very much for the kind introduction, and I would like to thank the organizer for giving me the possibility to speak and to present the work of my group to you. So I hope that everything will work. I'm not used to present in this format. I'm very excited. It's kind of weird to be sitting in my studio at home and at the same time speak to more than 900 people. So can you please confirm that you are seeing my screen?
Yes, yes, we can see your screen.
Thank you very much. So the work in my lab is focused on understanding how simple molecular components self-organize to form large and complex cellular machines. And we do that by focusing on a special organelle, which is called the cilium, or also called flagellum for certain cell types, which is an ubiquitous organelle that can be found in most eukaryotic cells, ranging from single cells, like you can see here, chlamydomonas or tetrahymena, which are two of the model organs we use in the lab, to cells of complex tissues in many organs and tissues in our bodies. So they have important motility, signaling, and sensing function, and the defect in the assembly and therefore the function of these organelles can generate a number of devastating human pathologies. Some of these pathologies are derived by mutations that affect the motility of the cilium, while others are instead caused by mutations that affect the signaling and sensing function of the cilia, and in this case, mostly of primary cilia, which are the non-motile cilia we have in our body. Now, the function of the cilium is performed by a very complex structure, which is called the cilium axonym. This structure is the main component of this structure is tubulin, which is forming a system of 9 microtubule doublets, plus, which are beautifully organized in a 9-fold symmetrical way, plus 2 microtubules at the center in the case of motile cilia. As we will see later in primary cilia, these 2 microtubules are not present. Now, every microtubule doublet contains a complete microtubule, the A-tubule, and an incomplete microtubule, which is called the B-tubule. And beside tubulin, in the cilium, we find a huge number of other microtubule-associated proteins or membrane-associated proteins, so proteins that are in the matrix of the cilium. And proteomic analysis of motile cilia shows more than 1,000 proteins. Now, usually these proteins are transported in the cilium in huge numbers, many of them, as I just mentioned, associated with the microtubules, and they do that with a very specific geometry by following specific repeats, and also by the specific filaments of the microtubule doublets. We can number the protofilaments in microtubule doublets simply because, in a simple way, because we have a symmetrical structure, and in a symmetrical structure where we have the A-tubule and the B-tubule. Now, how the cell managed to do that, how all these simple components managed to find the correct position on the microtubule doublets is actually not understood. what we know, so in my lab, we try to understand what are the molecular principles that regulate the transport of these components and also allowed all the components to find the right position in the structure of the axoneme. So what we know about the assembly of the cilium is that we need a basal body, so a template that is embedded in the cell body. And we also know, and was discovered in 1993, that the assembly of the structure happens at the plus end of the microtubule, which is located at the tip of the cilium, the distal part of the cilium. Now, the cilium can be very long, and diffusion is not an efficient way to move all these components to the tip of the cilium. So Joy Rosenbaum and collaborators in 1993 discovered that there is a specialized bidirectional transport system, which they called Intraflagellar Transport, or IFT, which is responsible for moving components of the cilium from the cell body where they are synthesized to the tip of the cilium where the assembly happens, and then moving the components of the cilium where the disassembly of the cilium happens, or signaling molecule back to the cell body in an anterograde transport and a retrograde transport. So the anterograde transport and retrograde transport, sorry, something happened with my presentation. Yes, okay. Anterograde transport is performed by a kinesin motor, while the retrograde transport is performed by a dynein motor, which are specialized for the IFT transport. And the interaction between the cargos, the many different cargos, and these motors that run along the microtubules is done by, is mediated by a two huge complexes of IFT adapter proteins, which are called IFT-A and IFT-B. IFT-A comprise 5 proteins, while IFT-B has more than 17 proteins. So we know quite a lot about the biochemistry of this system, and quite a lot about the fact that even mutation for single components of the IFT machinery can cause devastating pathologies. However, we still need to understand a lot of details about how IFT function and what are the molecular mechanisms that drive IFT. One thing that we were looking at when I started my lab at the Max Planck Institute in Dresden is we were looking at the dynamic of IFT trains, we call them, in the cilia of Chlamydomonas, also called flagella, in turf microscopy. And this is a very beautiful way to image IFT. You can see here in this movie, I hope that you can actually see them. I was told to optimize the screen resolution in order to allow you to see the particles moving in the flagella with the right frame rate. You can see that they move very quickly. They move up to 4 microns a second, up and down the 2 flagella or 2 cilia of Chlamydomonas cells. These are GFP tagged IFT trains. And we noticed that also, we have more than 20 trains zipping up and down the cilium in a given time. We never see collisions happening between trains that move in opposite directions. Sometimes we see, we manage to see trains that push each other in the same direction, but we never see collision happening in the opposite direction. So we ask ourselves, what is the mechanism that allowed the cell to regulate the transport system to avoid collisions? And there were 2 hypotheses that were present in the field. One hypothesis was that the trains were able to switch microtubule doublets before collision would happen. The other hypothesis instead was that the trains that there were microtubule doublets that were specific for different direction of movement. So we said, we want to address this question. We want to figure out what is going on here. And we knew that we could look at the dynamic in turf microscopy, but at the same time, we also realized that we will never be able in turf microscopy or in fluorescent microscopy with such a speed to resolve the intimate interaction between microtubules and individual IFT trains. Now, thanks to work that I have done in the past when I was still in Italy in the lab of Pietro Lupetti, I knew that using electron tomography or electron microscopy, it is possible to visualize IFT trains and also visualize their interaction with individual microtubule of the cilium. Therefore, we decided with Ludek Stepanek, my first PhD student, to combine electron microscopy and turf microscopy with a time-resolved correlative approach, and integrate the dynamic information, the high-resolution information together to understand how the trains use the microtubule doublets in the cilium. Now, the problem we figure out very quickly is that these trains are very, very fast. And the technique that were available at that time, they were not fast enough in order for us to catch the train, to visualize the dynamic in fluorescent microscopy and then catch the trains, the same trains in the EM. So we came up with our own method that works in the following way. We put a chlamydomonas cell or a number of chlamydomonas cells on a glass slide, and then you will see that we focus, in this case, you will see here what happened when we have the dynamic happening in one cilium. This is the base. So here we would have the cells, and this is the tip. You will see what happened to the trains if we plot space over time. And what happened is that we can track the position, the traces left by individual trains. And you see that anterograde trains go in this direction, retrograde trains go in this direction, and that suddenly all the trains become vertical, all the traces become vertical. This means that the trains are not moving anymore. And this is happening because we intentionally put fixative in the flow chamber where we are imaging the cells. This allowed us to stop the trains, and in this way have a fixed position for each single train, and also allowed us to go back in time and for each single train figure out if this was an anterograde train or a retrograde train. So using this approach, we were therefore, and then using associated image analysis, very simply taking the chymograph and doing a Fourier transform, masking different parts of the Fourier transform, and then using a back Fourier transform to select, in this case, for example, only the anterograde trains from the chymograph. We were able to color code all the trains we had in the chymograph based on the direction of movement. And don't forget that now the cells are fixed on the glass slide, so we can go on with our method and prepare the cells with what is called a flat embedding, which means that the cells are on the glass slide and we can prepare them for resin embedded for tomography. So we section in the first section that we get, the interface between the glass and the resin, we get all the cilia of all chlamydomonas. We look for our specific chlamyd that we were imaging. We perform a number of tomograms that allowed us to reconstruct a full cilium. And then we correlate, we do tomography on that. So I guess you are very familiar with what tomography is, so I can skip this part. So we rotate our sample in the microscope, we take different views, and then we combine them to generate, computationally to generate a tomogram. So by doing that, we managed to reconstruct, to generate a full reconstruction of an entire cilium where we can find all the IFT trains, the densities of them. We correlate this with the fluorescent microscopy, and at this point in time, we have not only the position of the train, but we can also see the morphology, on which microtubule they are running, because we are in tomography, and we know if this train was an anterograde or a retrograde train. So using this approach, we figure out that the anterograde trains, the one that are moved by kinesin motor, and retrograde trains that go back to the cell with the dynein motor have a different morphology. This was speculated for a long time, but this is the proof that this is indeed the case, and also shows that the trains reorganize at the tip of the cilium in order to have the two different functions, two different directions of transport. But not only that, now we can answer the question of how the trains use the microtubule doublets in order to avoid collisions. And we found out that practically all the microtubule doublets can be used in the cilium. We have a number of experiments done, can be used by both anterograde in green and retrograde trains, as you can see in this graphic. However, we also noticed that anterograde trains were associated with the B tubule of the microtubule doublets, while the retrograde trains were associated with the A tubule of the microtubule doublets. Now, this is how you would see an anterograde train and a retrograde train in tomography, in the tomograms that we reconstructed. We actually wanted to have, we could do averaging of these densities and have an idea of where the trains were. But we wanted to have a better imaging, and of course this is not possible with the type of cutting of the section that we made, because we have a missing wedge artifact in our reconstruction. Therefore, we decided to change the way we cut the sample, and use the salami slice technique in order to have the axoneme in a different orientation that would allow us to stitch a high number of tomograms and have a full reconstruction of the cilium without, where the missing wedge was in a position that didn't affect our ability to see the trains. So how we did that? That's very, in principle, very challenging, but Lude came up with this idea. He produced a PDMS stamp, probably mold, probably this is not the right word, but I guess you understand what I mean, on the glass, and then put cells in these compartments, a drop of cells on this mold, and image the cells that were single cells in this hexagon. Now, then he performed all the embedding steps on this PDMS format, and then remove the PDMS, identify the position of the one cells that we were imaging, remove all the rest of the embeddings that were just not interesting, reoriented the hexagon in the way that the tip of the cilium was pointing at the tip of this mold for the re-embedding the block. And in this way, it was possible to cut perpendicularly the cilia in a much faster and easier way. And this allowed us to visualize beautifully anterograde trains and retrograde trains in this new tomographic reconstruction, where we can even see the connection between the train and the B-tubule in the case of the anterograde trains, and the connection between the train and the H-tubule in the case of the retrograde train. So by using this approach where we combine turf and transmission electron microscopy at room temperature, we were able to discover that the collision between anterograde and retrograde trains is avoided just because anterograde trains move along the B-tubule while retrograde trains move along the A-tubule of each microtubule doublet in the cilium of Chlamydomonas. This also means that kinesin motor somehow managed to recognize the B-tubule, and dynein motor recognize the A-tubule. And of course, this is a bit puzzling because we still don't know what is the molecular mechanism that allow the motors to recognize the right track they have to work on. So we are at the moment in the lab investigating possible answer to this question. And one thing that we are investigating deeply is the role of tubulin post-relational modifications in marking the surface of the microtubules that can be recognized by the motors. And there were studies in the past where it was already shown that the A and B tubules bear different post-relational modifications. So this is a very intriguing idea. Okay, so this cartoon explains very well what happened in terms of separation, spatial separation of the tracks for anterograde and retrograde trains. But what you see here, the morphology of the trains, is practically a very artistic interpretation of what the reality is. So at that time, we didn't really have an idea of how the trains assemble in the cilium and how all the different components of the IFT machinery form a train. So we didn't know any much more than what we can see here from the room temperature electron tomography about the morphology of the trains. Of course, if you want to understand how the different components contribute to the structure and how they interact and how they build, the trains are built, we need to have better resolution. And for this reason, we moved to cryo-electron tomography and subtomogram averaging that, as you know, allows to gain much more high resolution insights into the structure in situ. So I guess you saw a number of movies about electron tomography. This is just the one specific for Chlamydomonas that we made. You can see here Chlamydomonas happily swimming. They get frozen directly on an EM grid, inserted a cryo-electron microscope, in our case a 300 kilovolts Titan halo microscope. Different views are acquired while we tilt the sample, and then these views of a segment of the cilium are used to computationally reconstruct a tomogram that can be inspected in order to look for IFT trains or microtubule doublets and all the proteins associated. And then we use subtomogram averaging. We pick particles that contain the same structure, align them, average them, and in order to increase the signal-to-noise ratio and have a better resolution. So this is a typical example of a segment of a cilium of Chlamydomonas that, and there's a tilt series. And this is the results after reconstruction. And in this kind of tomograms, it's difficult to see it like this on the fly, but you believe me, we can find IFT trains. And they are located, as we knew already from room temperature electron microscopy, in the space between membranes and microtubules, because some of the components of the IFT trains are associated with the membrane while the motor walk on the microtubule. And through the comparison of the results that we got from the correlative microscopy analysis and the cryo-EM, we were able to distinguish anterograde trains from retrograde trains also in cryo-electron microscopy. So from now on, what you will see, the results that I will present are related to anterograde trains. So if we zoom in the structure of an anterograde train, we find out that looking at different sections, we find out that the trains are built by three different kind of polymeric structure, where we see units or domains repeating with the same morphology. And this is an 18 nanometer repeat, a 6 nanometer repeat for the green one, and an 11 and something nanometer repeat for this yellow component. So using subtomogram averaging, we were able to improve the resolution of the model of these different components. And we, here you can see an isosurface representation of the average. And this is the first time practically that the IFT machinery was visualized in cryo-EM and in cryotomography as well in situ. So, I'm sorry for this. I don't know if you are seeing what I'm seeing here. So how do we move now from this 3D visualization to understanding the composition of the train? We use mutants that we knew were lacking certain components of the machinery. And in particular, we decided to use a mutant for the complex A, so the complex of adaptor protein that contains 6 adaptor proteins, and then a mutant for the dynein motor, which is the retrograde motor, the one that is responsible for the retrograde transport, which is supposed to be associated with the anterograde trains, because somehow the dynein motor needs to go from the minus end of the microtubule to the plus end of the microtubule at the tip of the cilium in order to be then performing the retrograde transport. So what we found out by comparison of the wild type structure with the comparison of the IFT, on the complex A mutant, is that this electron density here that repeats, this component here that repeats with an 11 nanometer repeat, are indeed complex A components. And this makes a lot of sense for the biology of IFT, because the proteins that compose IFT-A are known to be responsible for transport of membrane-associated proteins in the cilium. And as you can see from this illustration, these components is facing and interacting with the membrane. Now, the second mutant was the one where we didn't have the dynein motor. And as you can see here, this huge density on top, which is here illustrated in dark gray and blue, is missing in the mutant. So we identify the position of the dynein motor, which is not the active motor in this case, but it is the motor that is responsible for the retrograde transport. So here we are looking at an anterograde train. Now, I can tell you a lot of details about the stoichiometry, and then what is left here in the structure is the IFT-B, which we found out is the major adaptor complex of IFT, and we find out that this is actually the only really necessary component to form anterograde IFT trains that can move to the tip of the cilium, because in our mutants often we find, or sometimes we find, trains that can be built on simply and only by IFT-B. So we have this vision of the train formation where the IFT-B is the backbone of the structure, and practically complex A and the dynein motor are more or less like cargos attached to the backbone of complex B. So I can tell you a lot of stuff about stoichiometry and what the position of the different components mean to us in the field of IFT and so on and so on. But for today, I would like to focus on what we can learn about how motors of the IFT machinery are regulated. You might remember that the anterograde motors are moved by kinesin. Anterograde trains are moved by kinesin motor, and retrograde trains are moved by dynein motor. And already in the past, there was this question about how is the dynein motor moving to the tip of the cilium, because this is known to be a retrograde motor, so it cannot go to the tip of the cilium. Already by fluorescent microscopy, it was shown in the past that the dynein motor moves together with the anterograde trains, but it has been puzzling for a long time on how it is possible that both kinesin and dynein motor are associated to the IFT particles, because, and at the same time, the IFT transport is extremely processive. So there is no stopping or changing in direction of the IFT trains where they move to the base of the cilium, from the base of the cilium to the tip of the cilium, in a fashion like the one that we see in the cytoplasm, where we have both dynein and kinesin motor associated to the same particle. And this is usually called a tug of war. So we have no tug of war in IFT, but we know that both motors are associated with the same trains. So if you look at the structure of the motor that we find attached to the IFTB complex as a cargo, we find out that the motor is practically loaded on the structure of the train with a very tight repeat, 18 nanometers. And that the different motors interact with each other. So they are practically touching and being connected to each other. On the other, additionally to that, we see that the motor domain has a very particular conformation where the triple A rings are stacked and the stalks are crossed and interact with each other. And this conformation is very different from what we would expect for active motor that moves along the microtubules, where the rings are parallel and the stalks are disengaged from each other. This conformation was discovered after purification and visualization in single particle analysis already in the previous work by Carter lab and Roberts lab. And the Carter lab looked at the cytoplasmic dynein, while Roberts lab looked at a GST dimerized form of the motor domain of this dynein. Both of them identified this particular conformation and showed that the conformation is able to bind to the microtubules, but actually cannot processively walk along the microtubule. So what we discovered here is that the dynein motor, when it is loaded on the anterograde train, is inhibited by this particular conformation, which is called the phi conformation. And so we proved that this conformation is actually as a particular function in the cilium. Additional to that, the inspection of how the motor is loaded on the train, on the B backbone of the train, showed us that the motor is loaded in a very inefficient way if the motor was going to walk on the microtubule, because the tail is pointing, the tail is pointing towards the microtubule. The tail is the part that is usually associated to cargos, while the motor domain, that is the one that should walk on the microtubule, is pointing towards the membrane. So here we learned that we also have a positional inhibition of the motor, not only a conformational inhibition of the motor. So practically the motor is loaded on the train in the way that it cannot even interact with the microtubule. Now we were very happy to see that we have two different mechanisms that allow the motor to be inhibited during anterograde transport, the dynein motor. However, it's a bit puzzling, the fact that we have both, we have two different conformations, two different mechanisms. Why do we have such a redundancy? And so we think we understood why this is, why we have a positional and conformation inhibition. Now imagine that the motor was loaded in the way that the microtubule binding domain could still interact with the microtubule, so flipped the other way around with the stalks towards the microtubule. This would mean that the motor still binds to the microtubule, so it could slow down eventually the motility of the anterograde trains. So we need the positional inhibition to keep the microtubule binding domain away from the microtubule. But why do we need the conformational inhibition? So we look at the tip of the cilium, and we found out that at the tip of the cilium, we can catch trains that are disassembling and reorganizing at the tip of the cilium. What happened to the dynein motor is that the dyneins that you can see here in this zoom. Here you see the dynein motors in an inhibited conformation on an anterograde trains that is along the length of the cilia. You can see the two rings. And here you can see that the two rings of these different dyneins open. So they are not anymore in this inhibited conformation at the tip of the cilium. These particular dyneins that you see here are dyneins that are still somehow associated with the train. The train is in a disassembly stage, but they are still associated with the train. But we can also find this open dynein just at the tip of the cilium without any association with the train structure. So what we think is happening is that the dynein motor is released at the tip of the cilium in what is called open conformation, which is not yet active, but is disengaged from the train. And now if you imagine about, if you think about the role of the dynein motor is supposed to reach the tip of the cilium in order to bring the IFT machinery back to the cell body. So it makes a lot of sense that in the structure of the train, we have a auto-inhibited conformation, because once the train is arrived to the tip of the cilium and the dynein is released, this dynein cannot be activated, cannot be active immediately, because otherwise it would just walk back to the cell body without the IFT machinery. So the dynein must be in a sort of inactive conformation here that is activated only when the dyneins are already associated with the IFT machinery that needs to be brought back to the cell body. So of course, it's very interesting, will be very interesting in the future to understand what are the mechanisms that allowed this reorganization of the IFT machinery at the tip of the cilium, and also the activation of the dynein motor at the tip of the cilium. And of course, we are also continuing our investigation of what is the morphology and the structure of the different components of the IFT machinery, not only for anterograde trains, but also for retrograde trains. And we are using both single particle and tomography approach to investigate how the different proteins, adaptor proteins, interact in the two different complexes, and to identify the position of the many different cargoes that the IFT machinery is responsible for bringing to the tip of the cilium. Okay, so what I have shown until now was already published, and maybe some of you already saw these slides and some part of this presentation. What I'm going to show from now on is instead some new results that we have in the lab that we put on bioRxiv just a couple of weeks ago. And the motivation for this new work is the fact that most of the electron microscopy or cryo-electron microscopy in particular, so high resolution information that we have on the structure and the function of cilia, is derived from studies that use specific model organs such as chlamydomonas, tetraemena, sea urchin sperms, and so on. However, these are all motile cilia. And we miss completely to understand what is the structure and how this structure is related to the function in primary cilia. Now, primary cilia are very important organelles for our development and our health, and also for other vertebrates. And they are special cilia because they lost the motility function, but they have fundamental signaling and sensing functions that are responsible for many different, for the function of many different organs in our body. And so we have a variety of cilia in our body, and many of these cilia, as I said, are motile cilia. So why don't, sorry, just to tell you what we know from the textbook, the motile cilia that are the most studied, as I mentioned before, the structure is called the 9+2, because we have the 9 microtubule doublets beautifully organized in this symmetric way, and then we have the 2 microtubules at the center. Now in the textbook, if you look for the primary cilia, you will find that the primary cilia are called the 9+0 cilia. And this is due to the fact that we always thought in the past to the primary cilia like a simplified version of the motile cilia, where most of the components that are responsible for the motility function are just lost, and what is left is mostly these microtubule doublets organized in this 9-fold symmetrical arrangement. Now, from proteomic analysis, which is also a difficult task to do, of primary cilia, we know the primary cilia can be very specialized, not only for their function in different organs, but also for their composition. And we find components in primary cilia that are not known to be present in motile cilia. So we thought that we really definitely need to find a way to investigate the structure of primary cilia. And one thing that is important to mention is that the reason why we don't have a lot of information about the structure of primary cilia, and practically very little information about the cryo-EM structure of primary cilia, is that they are very difficult to isolate. So we decided to invest some time in developing a protocol that would allow us to isolate primary cilia from cultured cells. And for this development, we use kidney cells, ciliated kidney cells, which are very important for the function of our cilia. Mutation that affect the function of the cilia cause polycystic kidney disease. And the protocol is modification of what is called in the field as a peel-off, where we strip the cilia from the cell surface. And we do that directly on EM grids, and in particular for the cryo-electron microscopy on quantifoil grids. That we can freeze very quickly, and then in cryo-EM we can visualize primary cilia on the quantifoil layer. Okay, so we do tomography on these data sets, and already this is not cryo-EM, this is plastic tomography that we use to reconstruct a complete cilium. We learn that the architecture, the structure of the primary cilium is actually not conform to what we know from the textbook, where we was told that the microtubule doublets are organized in this 9+0 geometry. This was already observed by Sui lab in a different cell type, so it seems to be something that is recurrent in primary cilia. So what we see is that microtubule doublets are quickly migrating to the center of the axoneme. And so the 9-fold symmetrical arrangement is lost very quickly while we move from the cell body to the tip of the cilium. The other thing that we figure out is that most of the length of the cilium, 2/3 of the length of the cilium, is actually composed, and this is chopped here, so it's much longer, it's about 10 microns, is actually composed of simply singlets and not microtubule doublets. This is very unusual for cilia, and it's very interesting for us that are interested in IFT, mostly because, as you remember probably from the initial part of my presentation, we show that in Chlamydomonas, collisions between anterograde and retrograde trains are avoided by the fact that the trains can move in different microtubules of the microtubule doublets. So now, obviously, in the primary cilium, the trains need to reach the tip of the cilium, but we have only singlets. So how are the anterograde trains that are usually moving along the B tubule able to reach the tip of the cilium in primary cilia? So we had a look in our cryotomograms for IFT trains, and we were able to identify IFT trains, as you can see here, which were moving or associated with singlets towards the end, the distal end of the primary cilium. And of course, we have a very little number of these particles, so the average that we could generate is relatively low resolution, but we can compare the structure of the repeat of the B complex with what we had in Chlamydomonas, and see that also in primary cilia, IFT trains organize in polymers, so it's not a prerogative of Chlamydomonas, but it's something that seems to be conserved from algae to mammalian cells. And we see that these anterograde trains are able to move along singlets. So this completely changed our interpretation of how IFT trains are regulated, the logistic of the trains are regulated in mammalian cilia, and it will be interesting to see how this happens in other kind of cilia as well. The other thing that we observed and that we were puzzled about is the fact that, yes, we have mostly singlets in this primary cilia. And despite the fact that the primary cilia are not responsible for motility, so they don't need to be very, let's say, stable or rigid, like tolerant of deformation like the motile cilia are, they still need to withstand flow. Like we saw before in the kidney, the urine filtrate flow is impact on the cilia, and the cilium is responsible to sense this urine filtrate and the flow of the urine filtrate. So they need to have a certain rigidity. And so we were asking ourselves, how is this rigidity generated when we have very little or almost apparently no microtubule associated proteins on the outside of the microtubule and only singlets. So in motile cilia, we know that inside the microtubule doublets, we have a huge number of microtubule internal proteins, which are known to be responsible for stabilization of these microtubules. So we were looking for microtubule internal proteins in the microtubule of the primary cilium, and we found out that yes, we have some microtubule internal proteins, but definitely they are not organized in such a periodical and beautifully structured way as we see in motile cilia. And we were able just to, from the data we have, we were able to generate averages of particles that are associated with the internal wall of the microtubule, while we have also particles that are instead floating, seems to be floating in the lumen of the microtubule. So we were still puzzled about how is that possible that these microtubules are still stable in the primary cilium. And then we noticed in our tomogram that we have decoration along, I hope you can see it here, I hope the quality is good enough, decoration on the outside of the microtubule. And here is just a zoom of a raw tomogram where you see the density of the tubulin here and an extra density appearing every 8 nanometers on the outside of the wall of the microtubule. So we perform subtomogram averaging, and what we obtain is a density that has the dimension that would, let's say, fit with what we know for EB1 or microtubule end binding associated proteins, and also bind in between the protofilaments, but not in the space where we find the seam. So we were curious about the identity of this protein, and we decided to use immunolabeling. And we look for proteins that are known to be found in primary cilia. EB1 is one of these proteins, but in literature, EB1 is known to be associated with the microtubule plus end or the base of the cilium. So we were really surprised to see that our immunostaining instead in the MDCK2 primary cilia show that EB1 decorates the full length of the axonym, and this somehow resonate with what we have in our subtomogram averaging and our raw tomograms, where we can see this density associated with the full length of the singlets. Okay, the other thing that we found in the structure of the primary cilium that could also be, we don't know exactly what the function of this the last components or in unusual components of the axonema structure of cilia is, but could be associated also in this case with the stability of the axonym and also the regulation of the stability of the axonym depending on the needs of what the primary cilium needs to do, is actin filaments. So proteomic analysis has shown that there are, for a long time, that actin is found in the cilium. And also that there are several proteins that are known to regulate actin polymerization or actin activity in the cilium. However, the presence of actin filaments has been, not visualized, or anyway not known for in the literature for primary cilia or cilia in general. And actually we know that is not present in at least in the motile cilia of chlamydomonas. So we were surprised to find actin filaments in our raw tomograms. You can see one here. This practically is a bundle or a number. You can see here a number of actin filaments that are found in between the membrane and the microtubules, the singlets that we can find in our tomograms. And in some cases, instead, we find individual filaments that are intercalated between the microtubules along the length of the cilium. So we decided to perform subtomogram averaging, and here we see our low resolution reconstruction of a segment of the actin filaments that we compare with a structure that was already deposited in MDB. And of course, we wanted also in this case to validate this finding, and we use immunofluorescence. And here you can see cilia of cells that are forming a cyst, again, MDCK2 cells. And you can see here acetylated tubulin, and here you can see the signal of actin inside the cilium. Okay. So with this, I hope that I showed that this protocol that allowed us to strip the cilia, the primary cilia from cells, culture cells, is a powerful tool that can allow us to learn much more about not only the composition, but also the structure of primary cilia, which are very little investigated for technical reasons. And we hope that this will allow us also to investigate in the future what happened in cases where we have mutation that generate pathologies that are related, ciliopathies practically. So we will be interested also to expand our method, not only to MDCK cells, but to other kind of cells, ciliated cells, and at the end generate atlas of different morphology, different composition of cilia from the human body or invertebrates. So with this, I would like to thank you very much for the attention. I know that I'm the last speaker and this is the 1st of May, so everybody wants to go grilling, probably outside if the weather is good enough. But first, I would like, before closing the talk, I would like to thank my lab at the MPI-CBG in Dresden. I'm very happy to work with all these young people, and I would like to thank Mareike, Dennis, and Ludek for the work on the cryo-EM of IFT. And Ludek for the fantastic work he has done in developing the time-resolved correlative approach that allowed us to identify trains and their dynamics. And then I would like to thank Petra, Gonzalo, Nikolai, and 2 collaborators from another lab at the MPI-CBG, Alf and Ricardo, for the work on the primary cilium. And then I thank all the facilities at the MPI-CBG and all the funding sources. Thank you very much. I'm happy to take questions.
Thank you, Gaia. Thank you for a lovely talk. We've gone a little bit over time, but maybe we can shoot a few questions, maybe 2 or 3. So Meow Gui asks, do you have a density map for the retrograde IFT? And how is that different from the anterograde IFT?
Yeah, we have a density map for the retrograde IFT. Unluckily, the resolution of this density map is not as good as we have for the I'm trying to find you in my computer, but I don't find you. Okay, I trust that you are seeing me and that I can answer. So unluckily the resolution is not as good as we have for the anterograde trends, and we have some issues with some of the mutants. For example, the 2 mutants that we use for the study to identify the different components of the anterograde trains cannot really be used to look at retrograde trains because both mutants have problem with the retrograde transport. So we have a wild type structure, and this wild type structure looks different from what we have for the anterograde trains. So we have a different repeat, while in the anterograde trains we could distinguish 18, 6, and 11 nanometer repeat. In the retrograde trains, we have approximately a 40 nanometer repeat. And so the trains are much shorter than what we see, not much, are shorter than what we see in the anterograde trains. And given the fact that they have such a more spread periodicity, we estimated the number of components is also much lower compared to anterograde trains. And this agrees very well with what is found in fluorescent microscopy, where people were able, Ildit's lab in particular, I think, was able to demonstrate that when anterograde trains reach the tip of the cilium, then they split after reorganizing in 3 or more retrograde trains. So the retrograde trains contains a lower number of particles that we see also in cryo-electron microscopy. The repeat is different, and the way the complexes are arranged seems also to be different, because the morphology that we have is very different from what we see in the anterograde trains. But we don't have yet clear understanding of where complex B is and where complex A is.
Okay. Francesca Corsia asks, which post-translational modification do you expect on the two trains?
Oh, so, okay. So here I give you some information of stuff that we have done already. And I can tell you that polyglutamylation is not involved. We know that it's involved in regulating the activity of other axonomal components, but we find that it's not involved in the regulation of IFT, at least in Chlamydomonas. We are now investigating polyglycylation and detyrosination and tyrosination.
Okay, thank you. And maybe one last question from Joe Atherton. What software did you use for picking and processing your microtubules and actin filaments from your tomograms?
We use PEAT.
PEAT, okay.
From the ARMOD package.
From the ARMOD package. Great.
We have a limited number of particles, so for the moment, pushing so much in the image analysis is, let's say, not, yeah, in the future, it would be more interesting to use also a different approach, more sophisticated approaches.
So this was done on a Titan Halo with a side entry holder, is that right?
Yeah, with a K2 camera, a side entry holder. And I think there was no faceplate data that I showed you today. We have a faceplate, but we don't really use it for axonal structure. We use it for different kind of samples.
So there could be room for improvement just from a methodological side.
Right. Yeah. Some of the tomograms that you saw in the presentation were denoised. I believe I put a note there, and this was done with a software that is a neural network-based software that is called CryoCare. This could be interesting for somebody.
Great. There are more questions for you in the Q&A panel.